Oh, here we go. Hello? Do I have to do something?
No, I think you're in. Hi, can you hear us?
Hello? Can you hear us?
The picture is of...
Yeah, the picture is...
Yeah.
That's what we saw the last time. Maybe they're just still setting up. We're a bit early.
It could be that they've left the equipment on to be sure of not missing it.
We could have a lot of people, like, we could have very good people, so don't get discouraged.
That's fine.
Last time, some students were coming in and out, and no apologies for personally, but so there's nothing I can do about it.
And this switches it to... So now they would see that.
Yeah, now they would see.
Okay.
And you see what they
More or less what you're seeing there.
So that's not the only view. They have two cameras, and you can view like a single person talking, and you can also view the audience, and they switch it to the audience.
Okay.
They have like two different cameras.
But it's looking as if they're switched on and they're going to file in precisely at 12:30.
It is being recorded now by Edinburgh.
Yes, it should be. Yes, it's recording and live streaming now. Live streamers won't see anything hopefully exciting, but it is all on, which is a step.
This is Heliconius.
Heliconius, yeah, cool. Yeah. You just say butterflies.
I think maybe I'll just say butterflies.
We'll be talking about Heliconius specifically a little bit.
I'm trying to see how can I contact.
They're called into us. So there must be something.
It looks as though the machine's on and working, but there isn't anyone in the room.
No, there's,
Okay, let me see if I can code this.
Ah, message from Ramiro saying there's no sound because it's muted, but
There's no sound because it's muted.
he's muted it at his end, so presumably they're not ready yet. Shall I email him back saying, are we going to start fairly soon? Or
Yeah.
Or do you want to email him?
If he,
Sure.
I can email him.
just,
Yeah.
yeah,
Well, he's just there. He's busy.
maybe email him and ask him
What do you want me to ask?
when to proceed.
But why is he muted it?
I could just,
He is in the email. He's using his email, the email I sent you?
Yes.
Okay. You're sending him an email already?
Yeah, I'll say we're ready.
Something.
Last time they were there.
see somebody there.
Yeah, there are shadows and.
They're gone. Someone's gonna listen to yourself.
They're probably waiting for everybody to come in.
Yeah, but they have to tell us that, something. Did you feel in the building the papers were demolished? Did you feel it in the
No.
We felt it in the floor. It was like
Y creo que aquí hay que mover esto de acá.
Hey.
Ok.
Ah, y entonces para yo verme, que sabes que no estamos viendo.
Ok. Entonces, pasa que el control cuando tú lo pones se activa esto y quita la pantallita. Pero para tu verte, yo te lo preparo. Si yo cogí todas mis clases con esta cosa.
Ah, ¿qué se coge con todo?
Ok.
Ahí estamos en la cámara.
Es la. No, espérate. Quito que se vea en el... ¿Te acuerdas que te apretaste que cambiaste la cámara? Y pues no necesitas que se vea ahí, sino que apretaste Snapchat.
Sí.
Ahí está.
Ahí tenemos cámara. Entonces,
Y con ya puedo mover la cámara por aquí.
ahí está.
Hello.
Hello.
We can hear you.
Yes, I can hear you too.
Okay, great.
Entonces, ¿cómo?
So, está viendo, no me está viendo. Está viendo a Martín. Martín.
Sí, tendría que mover para acá.
Vamos a ir para allá.
No, ya está bien, es que la cámara no está moviendo.
Ah, okay. Ahí estamos. All right, hi, I am.
Great.
You already hear? Yes, I am Juan Carlos Martinez, and we have several group of students here, ready for the webinar.
Wonderful.
Huh.
Again, I'm just going to do the introduction. This will be our second of 5 webinars that we have scheduled now. I just want to bring up our website. If you have a topic that you'd like to hear about and any suggestions about that, you can use the contact tab to send an email to us, and we'd be happy to hear about any topics that people would like to hear about. And if you have a chance, we have a survey. If people have any feedback, we'd be more than happy to hear how you guys are enjoying these webinars. And you can follow us, updates on the next coming webinars here. And also you can follow us on Twitter, Instagram, and Google+. And today our speaker is John Davey. John was a research fellow, is a research fellow here in the University of Cambridge in the Department of Zoology, and he's studying the evolution of butterflies. Previously, he was at the University of Edinburgh, where he worked on rad sequencing, bioinformatics, and other next-generation sequencing topics. And the title of his talk today is, "What can DNA sequencing data tell us about non-model organisms today?" So, John. Yes, of course.
All right, good afternoon. Thank you for coming today. I was wondering if you could swing the camera around a little bit just so I can see the audience. That's great, thank you very much. Hi, thank you very much for coming. Thank you to Paul and to Monica for inviting me to do this. It's a great pleasure to speak to you. I'm going to just talk for maybe about half an hour, 40 minutes about what we can do with DNA sequencing for the kind of questions that I think interests many of us. And I'll say exactly what I mean by that in a few moments. So, genomics is big news. There's a huge amount of coverage about what's going on in DNA sequencing and other kinds of sequencing, and particularly around what we can do with human genomes. It's only about 13, 14 years since the human genome was published for the first time, and there's been enormous progress since then. We're already talking routinely about sequencing genomes for about $1,000. It's in this book by Kevin Davis from a few years ago. So that's not much more than a new iPhone. And Illumina, which is the company that does a lot of the sequencing, provides most widely used sequencing machines, has just announced that they believe that there will be 228,000 human genomes sequenced this year. And so what I'd like to go into is what that really means, what we mean when we say we're going to sequence 228,000 genomes this year, and what the implications are for us who maybe are not working on humans. So this progress has been very real. These numbers are not entirely hyped up. there has been an incredible drop in the cost of sequencing over the last 15 years, from about $100 million back when the first human genome was published, to somewhere between $1,000 and $10,000 today for full coverage of the human genome. That seems to be leveling off a little bit. We'll talk a little bit about that later on. But the rise, the fall in cost has been enormous, this is a real thing, and that's making huge differences to what we know about many human diseases. We have very, very good information about variants for almost any disease you care to name now. It's less clear what those variants actually mean, and there's still a huge amount of work to be done to work that out. But in terms of sequencing the variants behind particular diseases and thinking about how to introduce that into the clinic, that work is well in progress. But I'm assuming that that you in the audience today, and for me also, we are more interested in non-model organisms. Things like the Puerto Rican parrot, which I'm sure you know, it's just had its genome sequence published recently. Things like coral reefs, things like cacao, and things like the heliconius butterflies. So either critically endangered species, or ecologically important species, or agricultural species that may be neglected, or topics in evolutionary biology in general. What all of these have in common are species that have not routinely been worked on at a genetic level. And so the question is, what can we do with these species with the new sequencing technologies that we have available? So I'm assuming that the kind of questions that we might be interested in are things like this. We might be interested in understanding the genetic basis of a whole set of traits for animals that we are working with. So, for example, here's a study that's been published last year on the lake whitefish, which had part of its genome sequenced. That whole populations of these fish were worked on. Their genomes were sequenced to some extent. We'll talk about exactly how it was done later on. And they were measured for a whole range of different traits, the behavioral traits, morphological traits, physiological traits, things like growth rate, things like direction of swimming, all kinds of different things like that. And it was possible for these researchers in one study to identify the loci underlying most of these traits across the genome. So they were able to build a linkage map of the whole genome, the 40 chromosomes of the whitefish that you can see here, and they were able to place many of these different traits onto the chromosomes. And you can see these colored bars. Each one of these is some kind of phenotype, and the genetic loci that underlie those phenotypes are marked on the map here. So that's a huge advance on what people have been able to do in previous years, and I'm assuming that we're interested in doing things like that in species like coral reef, in species like the Puerto Rican parrot. I'm also thinking about things like phylogenetics, so we might be interested in the history of a species or a set of species. And here's an example of this. This is the pitcher plant, and it's actually the pitcher plant mosquito that we're interested in. So the black mosquito is hovering in the rim here. This is a mosquito that is a symbiont of the pitcher plant. It feeds on the bacteria and the other smaller insects that fall into the pitcher plant, into the water that sits in the pitcher plant, and it breaks up those insects, breaks up the bacteria so the pitcher plant can digest them, as well as eating something for itself. And it lays its larvae in the pitcher plant, and that's how it breeds. So there's a symbiotic relationship between these two creatures, but obviously quite an esoteric study system. There's several groups in Oregon, including Bill Bradshaw and Christine Holtzapfel, who are interested in the history of this species and its behavior across the northern United States. And you can see here a map of locations of all of these different populations of the pitcher plant. And so we want to know where these pitcher plants have come from. What are the origin sites for these populations, and which are the dispersed sites? And so this has been done several different times over the years. It has been done with just a single mitochondrial gene in the past, and you can see that it's possible to group these populations into clades here, but not really resolve some of the clades within that. Now, with the more modern sequencing technology, we can use many markers across the whole genome and actually get a very, very well resolved map of the populations. And so I'm assuming we're interested in maybe doing this for some other more local species. And finally, we may be interested in doing population genetics. So we want to think about the history of these populations and how they may be speciating, how they may vary within particular populations. And here's an example of some work done by Simon Martin and Chris Jiggins here in Cambridge. This is the group that I'm working in now, and various other groups, to do with the heliconius butterflies. These are just a few of the species of heliconius from the northern part of South America, which Ricardo Papa works on in Puerto Rico as well. Hello, Ricardo, if you're there. And this is work that we've been doing over the last few years, sequencing whole genomes of these butterflies and looking at the diversity between different species. So I'm not going to go into the details of this plot, but this is showing a measure called FST, which measures the diversity between populations compared to the diversity within populations. And it shows that within closely related species, there is very, very little difference in these butterflies, or even within one species, there's very little difference, except at particular loci. So you can see here, there's a big difference, and here there's a big difference. Those happen to be the loci controlling the color patterns between these two species. So it looks like it's just those two loci that distinguish the race of those species. Whereas if we look a bit further afield between, for example, Heliconius sydno here and Heliconius melpomene, two very distinct species, there's a lot more difference across the whole genome. So these kind of surveys are now possible even in a fairly non-model organism, and I'm assuming we're interested in that kind of thing as well. So what are the technologies that we can use for doing this kind of sequencing? So this has been a very vibrant field over the last 5 or 6 years when this big expansion in sequencing has really taken place. And basically there's been a single winner in that field. So we've gone from having maybe 4 or 5 viable companies at any one time to really just having one company that's doing most of the sequencing in the world right now, which is Illumina. And Illumina provide 2 widely used systems. They provide various others as well, but these are the 2 that people tend to use. One is the HiSeq machine, the other is the MySeq machine. The HiSeq is more of a large-scale machine that you might find in a sequencing center or in a department, a bit like a confocal microscope or something like that, whereas the MySeq is a bit more like a PCR machine. It's a bit more like something you'd have on the benchtop, although it's quite a bit bigger than a PCR machine. And cost quite a bit more too. But these machines are capable of sequencing many times coverage of the human genome. So for one sample, if you ran one sample on a HiSeq, one human sample, you might expect to get back about 10 to 15 times coverage of the human genome, as in 15 human genomes worth of data, for around £2,500. Now these figures are very, very rough. You will find huge disparities in this at different sequencing centers, but it's just to give you a rough feeling for what's possible now. On the MySeq, you get a bit less coverage, about 3 times coverage, for less money, about $1,500 US dollars. So this costs actually quite a bit more than the HiSeq, but it's a bit more convenient, and you might be able to do it within the lab rather than having to send your sample off to sequencing centers to do it on a HiSeq. But does this actually mean that we get out a whole genome? Is this what we mean when we say we can sequence 15 human genomes or 3 human genomes? Well, not quite. Because what we actually get from these machines is not whole chromosomes, is not whole genomes in one long strand, but we get millions and millions of short sequences. So with the HiSeq, we get 100 base pair sequences, so 100 letters in each read. With a MySeq, we get about 250 base pairs, which is quite an improvement on 100, but it's still a very small bit of sequence. And so we can generate 15 times coverage of the human genome, but it's all in 100-letter pieces. And so that's some distance from having a complete human genome, and we'll think about the implications of that in a few slides' time. The other machines that are coming up, which just briefly, oh, sorry, so one more thing about that. So if you have these short reads, this is an example of the kind of data that you might have. So what we're looking at here is a stretch of the E. coli genome. So this left to right is a stretch of the reference genome of E. coli. E. coli, of course, already sequenced, and the genome is well known. And these grey lines here, piled up against the reference, are individual reads that have come off one of these Illumina machines. And so we can pile up these reads onto the genome, and we can see how the samples that we've sequenced differ from the reference. And so hopefully you can see here that we've got this blue line here, that's these Cs, which differ from the reference genome, which has a T in red down the bottom here. And so by sequencing a genome and aligning it to a reference, we can call variants, we can call single nucleotide polymorphisms like this, and we can think about how those samples have differed from our reference strain. Two machines that are coming up and that will probably be very important in the years to come are the Pacific Biosciences machine and the Oxford Nanopore machines. The major selling point of these machines is that they produce much longer reads. So rather than the 100 base pair reads that we're getting from Illumina, the Pacific Biosciences machine will produce about 5 to 10 kilobases at a time, so 5 to 10,000 bases. It looks like the Oxford Nanopore machines are going to produce about the same length of sequence. The Pacific Biosciences machine will produce you about 1 times coverage of the human genome for about $4,000, so that's quite a bit more expensive than Illumina, but you get these much longer reads out of it. It's unclear how much the Oxford Nanopore is going to cost because this is still in early access at the moment, it's still in testing, but it looks like we might get about 1 times coverage of the human genome for about $6,000 or something in that region. You can see there is a vast difference between these two machines. The Pacific Biosciences machine is enormous, but you can see how huge it is compared to the size of a person here. This is the Oxford Nanopore machine down here, this little USB stick hanging off this laptop. This appears to work. This is not science fiction. It really is a case that you can put in a DNA sample into this thing and get sequenced straight into your laptop. As I say, this is still early access. It's not commercially available yet. Hopefully, maybe early next year, it will be available to us. When Oxford Nanopore first announced this machine, they said, you'll just be able to drop blood in here and get sequenced straight out. So the idea was that you could go out into the field and collect something and sequence it there on the spot. Now the machines are actually available to people. It turns out that there is a library prep involved. You do have to do some ligations onto your sample, and so it's not quite as straightforward as that. You will need a molecular biology lab to use this machine, but it's still extremely exciting to think what we might be able to do with these machines in the future. The only drawback with these machines is that the data is a lot less accurate than the Illumina data. So Illumina data is not perfect. You still get some error in the reads, and we'll see the implications of that in a moment. But with these longer read technologies, there is actually quite a lot of error, maybe between 11 and 15%. So that is, in every 100 bases, maybe 1 in 9 bases is wrong. So that's quite a problem, and we have to think very carefully about how to handle that data. Okay, so those are the technologies we have available to us. What can we actually do with them? This is the problem space that we're working with. When we think about the genomes that we're interested in, so a human genome is about 3 gigabases in size, but the human genome is just one of millions of genomes, just as humans are only one of millions of organisms. And actually there's a huge range of different genomes that we might be interested in. Here's a figure that shows genome size in base pairs, in DNA letters, on the log scale, on the x-axis down here, ranging from about 100 kilobases to 1 terabase up here. So here's 1 gigabase here, about 1 billion bases. And here are the ranges for different types of animals. So we've got humans up in the mammals here, about 3 gigabases. So that's bigger than all birds, but it's smaller than all salamanders and lungfishes and so on. So you can see there's a huge range of different genome sizes, and actually many of the very interesting genome sizes are enormous. For example, most gymnosperms and angiosperms, so lots of trees and plants that we might be interested in, have enormous genomes. Lots of fish have very large genomes on the scale of billions of bases. And so there are very many ecologically important and just generally biologically important genomes that we would like to study, but are very, very difficult to work with and very, very difficult to sequence. So thinking about the machines that we've got available to us, if we sequence a genome on one of these machines, as in if we take a DNA sample from a species that we're interested in and sequence it on one of these machines, we are not likely to get back a nice karyotype like this human karyotype here, where we get 23 pairs of chromosomes, and each chromosome is tens of megabases long, tens of million bases long. And what we're going to get back is an incredibly difficult jigsaw puzzle. We're going to get all of these 100 base pair sequences, and we're going to have to string them together into biologically interesting sequences like chromosomes or like gene sequences. And the problem is even worse than it looks, because genomes are mostly made up of repetitive content. The same sequence is repeated over and over again, and many of these repeat sequences are longer than the 100 base pairs that we can sequence with an Illumina machine. So, for example, in a human genome, there are many, many different types of repeat sequences. Two of the most common, for example, the lines, there are about 500,000 5 kilobase sequences in the human genome, which are pretty much the same. There's some variation in different copies, but they are recognizably the same sequence. And there are 1.5 million 300 base pair sequences called signs. So again, enormous quantity of these very, very repetitive sequences. And if we've just got 100 base pair sequences, we are not going to be able to read through these sequences that are actually quite a bit longer than 100 base pairs. What we're going to get instead is these huge pileups of repetitive DNA, and we're not going to be able to figure out how to sort these out, how to assemble these into long sequences. So this is a major problem, particularly as the genomes get longer, because we don't expect to find many, many more genes as we go to these larger and larger genomes. We expect to find roughly the same gene content relative to genome size, sort of 15, 20, 30,000 genes. We just expect to get more and more of this repetitive stuff bulking up the genome. And so the longer your genome gets, the harder it gets to assemble. Also, with some particularly hard plant genomes, particularly unfortunately it seems many crop genomes, you may have many copies of each chromosome. So in humans, we're actually somewhat fortunate that we've only got 2 copies of each chromosome, which is a difficult thing to address. It means that any batch of sequence, we've got 2 copies of that sequence with some variation, and we need to figure out which copy a particular read comes from. But in wheat, for example, we have 6 copies of every chromosome, winter hexaploid genome, and so we've now got this incredibly difficult problem where we've got 6 copies, and each one is gigabases of data. And all we have are these little jigsaw pieces. So the question is, how are we going to handle this? Because there's no question that if we could snap our fingers and have whole genomes of our populations, of all the coral reef organisms in a particular ocean, or many, many different cacao plants in a field or whatever, we would take them. We would take that data and work with it, and that would be wonderful. But if it's going to take us months or years or decades to put together these genomes from this short read data, what are our alternatives? What can we do instead of doing these genome assemblies? And we actually have quite a few options available to us which we can use to answer many of our questions in conservation and ecology without having to have these whole genome sequences. And so I'm just going to run through 3 possibilities that we might want to consider: RNA sequencing, targeted enrichment, and RAS sequencing and related genotyping by sequencing technologies, and then show some applications of those technologies. So, RNA sequencing first. So actually, arguably, a lot of what we're interested in is the genes in a genome. We don't need to sequence all of the repeats. That's probably not got a huge amount of useful biological information about the traits that we're interested in. It may well be sufficient to just get the gene sequences and compare those between different populations. And so one of our options is to take an RNA sample from a species of interest and sequence that. And if we do that, we could either try and assemble those transcripts separate from a genome. So let's say we don't have a reference genome, we can try and assemble those transcripts on their own. And that can work well for many genes. Or we can try and align those genes to some reference sequence we might have available. And that would get us pileups like this. So here, again, these grey bars are reads that we've aligned to a reference. And you can see here we've got 4 different exons across this gene. And this is just an example of the kind of variation we might want to look for. This is a heart sample at the top here, a liver sample at the bottom here. And you can see on the right that the heart sample has a different exon 3 to the liver sample. And so that might be sufficient for us to differentiate between 2 different species that we're interested in. That might give us a handle on what's going on in different populations. And so that's one option that we have that can work well. Another option is a targeted enrichment process. So this would be where we know of a sequence that we're interested in, and we might want to just sequence that region of the genome. So for example, in the butterflies that we work on, we know that we have a couple of loci that control the color patterns in our butterflies, and we know what those sequences are in some species. So if we could just sequence those regions, that might tell us a lot about color pattern evolution. And so one of the methods for doing that, which I'm not going to go into in detail, but just to give you the principle, is the Shore Select method from Agilent. There's also the Nimblegen method from Roche that you might want to use, which has... Should I? Average check, there we go. Oh, no, I don't have control over this. Sorry about this. Okay, there we go. Sorry about that. So one of the methods we might want to use is the Shore Select method, where we take some genomic DNA that we're interested in, and we take a set of RNA baits. So they are sequences that we've synthesized to match the region that we're interested in. And we bind those baits to biotin. We then bind the baits to our genome sample of interest, and we use some streptavidin-coated beads, magnetic beads, to pull out these baits. So the streptavidin binds to the biotin is bound to the bait is bound to our genomic DNA of interest in our samples of interest. So we only pull out those regions of the genome that we're interested in. And if we do that, we can get data like this. So here again is a pileup of reads against a portion of referencings we might have. Here in grey at the top is the bait sequence. So this is the sequence that we're interested in, and here are the baits that we've designed that sort of overlap and are designed to map across this whole region. And then here at the bottom is our actual sequence, these big stacks of data that pile up against our baits. And where we haven't designed baits, we just get a little bit of sequence, but not enough to do anything with. So we really focused our sequencing just on the bits we're interested in. So that can work well, but again, we've got a sort of chicken and egg problem in here, which is how do we get to this sequence? If we have no information about our species of interest, if we have no genetic information whatsoever, how do we even get to this point? And actually, what's happened in the last few years is that there have been a range of new techniques that have been developed to address this problem without having to sequence whole genomes. I'm going to talk about one of them that's most familiar to me because it's something I've worked on for a few years now, called rad sequencing. There are lots of variants of this method, but I'll just give you the principle behind it. Have a look at genotyping by sequencing, GBS methods, if you want other variants. So the principle of these methods is to use restriction sites. So rad sequencing is restriction site associated DNA sequencing. And I'll just briefly talk about what a restriction site is and why they're useful to us. So restriction site such as EcoR1 here is something that cuts a particular sequence in the genome. So for example, EcoR1 recognizes the site GAATTC, and wherever it sees that site in the genome, it will cut the genome up and leave an overhang like this, leave these sticky ends hanging off our fragments. So if we take a genomic DNA sample and we digest that sample with EcoR1, react the sample with the EcoR1 enzyme, we will end up with a whole bunch of digested fragments in our sample. So here's an example where we might have two samples of interest, and each of these red blocks here is a restriction site, let's say an EcoR1 site. We can digest that sample down, and we'll end up with all of these different fragments in our sample. And these enzymes have been used for an enormous number of different things. I'm sure many of you are aware of them already. And they've been particularly useful for genotyping like this, for understanding genetic variation. Because we might expect to have, for example, variation in restriction sites between different samples. There may be a difference in the sequence here in sample 2, let's say, so it doesn't have a restriction site here. And so we get one long fragment from sample 2, whereas we get two shorter ones in sample 1. But we also expect to have variation downstream of the restriction sites. And that's what we're going to make use of mostly when we talk about rad sequencing today, and actually sequencing around these restriction sites. So let's see how that can work. Let's say we had our chromosome here, and there are restriction sites all the way along here that we're going to cut at. If we cut up one of these sites, what rad sequencing is a process of taking those fragments, making up an Illumina library that just contains the fragment either side of the restriction site, and then sequencing either side of the restriction site using a normal Illumina sequencing platform. So we end up with these 100 base pair reads, normal Illumina reads, but it's just the region downstream or upstream of the restriction site. And I'm not going to go into the details of how this is done, because it's quite involved, the process of it, but just to say that this is how we would, this is the kind of data we would end up with. We can also sequence the other end of a fragment, so if we shear these fragments down to a sort of size of about 500 base to 1 kilobase, we can sequence the other end of these fragments and get a whole range of different reads at the other end of these fragments. And so we can span a region of about a kilobase around the restriction site, and we end up with a pileup of reads like this. So again, each of these grey sequences is a read, and so we can get these pileups at a particular region, and again, call variants just as we would do if we had whole genome data. So treat this as whole genome data, call variants, and use those variants to assess what's going on in our population of interest. I'll just give you a concrete example of this, why we might want to do this instead of doing whole genome sequencing. We're working on this butterfly, Heliconius melpomene, which has about 300 megabase genome, so 300 million letters, 21 chromosomes. If we sequence that on a HiSeq machine, we would get about 100 times coverage of the whole genome from a single run or a single lane of HiSeq. But although that gives us 100 times coverage, because there is error in the sequence, because we need multiple copies of the reads to really be confident about the data that we're seeing, we can probably only do accurate scoring of variants for 3 or 4 butterflies in that data. So that's good, that's fine, but that means it's going to cost us about $500 to $700 for each butterfly, which is quite a bit of money. with RAD sequencing, let's say we chopped up the genome with a restriction enzyme called PST1, which cuts at a slightly different site to EcoR1. There are about 27,000 PST1 sites in the genome, spaced about 10 kilobases apart, but we're only sequencing these small regions around the PST1 sites, so throwing away the rest of the genome. So this gives us genome-wide coverage, but at a fraction of the cost of the whole genome sequencing. And we can get about 2,700 times coverage of the RAD sites in the genome, the PST1 sites in the genome, in one sample of high-seq sequencing. So that will give us accurate variants for about 100 butterflies. And now we're really able to do serious population studies. Think about doing things like association mapping, think about doing linkage mapping, think about doing population genetics and phylogenetics. And so this has really become a viable strategy for many non-model organisms where whole genome sequencing isn't really a realistic possibility. All the examples I've shown so far have been of aligning to a reference genome. Okay, so all these pile-ups, these grey bars, have been piled up against some reference that we already know. But actually, there's been quite a bit of work done to try and use these RAD sequencing data without a reference to infer what the loci are de novo, so with no other information. And in particular, there's a great program called STAX, which has been developed by Julian Katchen at Oregon, which tries to do this. So we end up with tens of thousands of candidate loci across the genome, which we can use for all of our various questions. But that's going to have the same problems as genome sequencing, isn't it? Because we're still going to sequence all of the repeats. There's nothing to say that restriction enzymes won't chop repeats, just as we would get repeats in whole genome sequencing. So can we really reliably do anything useful with these bits of genome, these restriction sites? Well, yes, we can. And the way we do that is by being quite severe about our biological criteria for selecting these loci. I'll just show you a few examples fairly quickly. Basically, the examples we've already seen. This tree that we saw earlier on, the pitcher plant mosquito, that was built with RAD sequencing. So these genome markers, these nuclear markers, were done from RAD sequencing data. And all we do is select the markers that are reliably assigned to one population or another and just throw the rest away. And actually, we don't need that many markers. I think a few thousand we used here, but there were many more thousand that were just thrown away. They weren't needed to build a tree like this. Similarly, here is a linkage map of the zebrafish, which was built by Julian Katchen, the writer of STAX. This map at the top here was built with a huge amount of labor using microsatellite markers. There are about 100 markers on every one of these chromosomes here. But on the RAD map, which was done in one experiment, we have 300 or 400 markers per chromosome. But again, just a few thousand markers, and the rest of the data can be thrown away. We've got a great linkage map, in fact, almost better linkage map than was possible with previous data methods. So, and we can be quite robust about which markers we choose because we can easily see segregation in a set of offspring and throw away the rest of the data that doesn't match that segregation. Similarly, these are a couple of projects that I've worked on, which sorry, as I'm running out of time, I won't go into in too much detail, but just to say that in the Atlantic salmon and in various snails, we've been able to identify candidate loci for phenotypes that we're interested in. So disease resistance in the salmon, snail banding and chirality, so left-right asymmetry in the snails. And really, we ended up only finding about maybe 10 candidates of interest just by looking at markers which segregated according to, for example, left curling snails and right curling snails, and then throwing away all the other markers, all the repeat stuff. We didn't need it. We had the markers that we needed to think about our loci of interest. So this kind of stuff is possible with these RAD libraries, and it leads to studies like this. The whitefish study that we saw earlier, this was done with RAD sequencing. These whole linkage maps were built with RAD, and all of these associations were produced by RAD. There's no whole genome, and as far as I'm aware, there's no other SNP chip data or anything like that that's gone into this. I'll skip over that. There are limitations to this data. The closer we get to ecological questions, to really sensitive questions like population genetics, where we really want to know what's going on locus by locus, this gets harder and harder to do with RAD data. And this kind of population study that we saw earlier, this is probably not going to be feasible with RAD data without a reference genome. When we're thinking about how much does a population diverge from another population, we really need to be confident that we are having a fair and unbiased representation of the data in our sample. And that's quite a challenge to come to with the RAD methods that we have today. We can produce the data, but we don't know if we can really identify the accurate genes from that, the accurate markers from that data. So, so I'm running out of time, so I will just briefly caveat this. I've hopefully shown that there are quite a few things that we can do without sequencing whole genomes, just using a method like RAD. But there are 4 caveats that I'd like to say so that we don't get too carried away with this. One is that I haven't talked at all about doing DNA extraction or preparing the DNA for sequencing on these machines. And these things can be difficult if you're planning on doing a project like this yourselves, or you would like to think about this in the future. You need to be a reasonably good molecular biologist to put together a library that will work on these samples. And if you're working on small animals and rare specimens, then you may well need to work on DNA extraction to make sure you have very good quality DNA, or very long DNA if you want to do something like specific biosciences sequencing where we get these long reads. Second important message is that manual analysis of these data sets is impossible, and that bioinformatics is essential. We are now thinking about data sets that are tens of thousands or hundreds of thousands or millions of variants in size. It's just not possible to go through them one by one and eyeball them. We have to think about ways to do this at the computer and ways to process this. So it's essential that you have the ability to write programs or to use programs that will analyze this data, or that you need people in your lab who will be able to do that with you. The next thing is, we can only actually go so far with these projects. So even these RAD projects where we were able to get nice results in terms of candidate markers that said this looks like this is the chirality locus in the snails, or this looks like it is the right organization for these pitcher plants, for example. That doesn't necessarily mean we've proved our case. And in terms of, so for example, with the snail project, we proposed some candidate markers for the chirality locus 4 years ago. We are still in the process of proving the gene is actually in that locus. We had sort of 4 or 5 candidate genes within the region, and it's taken us years to figure out which gene was the crucial gene. So it doesn't. It gets us a long way to understanding our problems, but there's still going to be all of that follow-up work to do. And that's true generally. Things like the Lake Whitebait project, identifying the genetic loci is one thing, measuring all the phenotypes is quite another. Understanding what's going on with the ecology of our species, what's going on across a whole continent or something like that, that remains a huge amount of work. And so even though sequencing is becoming a much easier thing to handle, all the rest of the biology is still a huge challenge and still something we need to think about very, very carefully. And so with that slightly pessimistic note, I will leave you and say thank you very much for your time and your attention. I'd be very pleased to take questions. Thank you very much.
I have a question.
Yes.
Hi. I would like to use RAS sequencing. How could I decide on which enzyme should I use? Which restriction enzyme should I use?
Yeah. So the enzyme. Yes. So sorry. I didn't cover too many of the technical details like this because I was assuming the audience was going to be quite general. But if you have questions like this, then please do ask. So the choice of enzyme is there are various things to think about. One is the amount of markers you need. So if you only need a few hundred markers, you can use a very infrequently cutting enzyme, so like an 8-base long enzyme or maybe longer. If you really want to get very good resolution of your genome, you might consider a 4-base enzyme or a 6-base enzyme to do that. One of the things to bear in mind is the library process with RAD. So you need to bind on adapters to your DNA fragments so that the Illumina machine will understand the sequence. So you bind on Illumina adapters, and those adapters bind onto the Illumina flow cell, the sequencer, and that's how your library gets sequenced. The way that RAD prep works is you have to synthesize adapters which contain the restriction site overhang that you're interested in. So if you're using PSD1, say, you will need adapters which have TGCA on the end of them. And that can limit your choices because it means every time you use a different restriction enzyme, you have to buy a whole new set of adapters. And if you're trying to do 96 samples at a time or something like that, that can be quite expensive. So we have tended to use PSD1 or SBF1 just because they share the same adapter sequence. The other thing about that is you might want to compare data across multiple experiments. So if you sequence one population with PST1, it doesn't make a huge amount of sense to do another population with EcoR1 because they won't be comparable data. So once you're locked into one enzyme, it often makes sense to stick to that enzyme. There are various programs available now where you can test your sequence of interest, or if you have a related species, like a related bird or something, if you're working on the parrot, where you can simulate your RAD data. You can take an enzyme sequence and say, right, how many cut sites am I likely to have? How many genes am I likely to cut? That kind of thing. So there are those things out there. It's worth having a look at those if you're still worried. Does that answer your question? Is there anything? Yeah. Okay. Okay.
I also one question on target enrichment.
Mm-hmm.
You showed us a figure there showing the baits you used, and one of them seemed not to have gotten much data. I imagine some baits actually are not have a sequence that may not be conserved, and they will miss their target. Wouldn't be missing the most interesting data by using target enrichment?
Yeah, possibly. Absolutely. So that is a risk with that method, that if you design to a particular sequence and you're interested in species which are some distance from that reference, then yes, you risk missing those important bits. The same as with a SNP chip or something like that. In fact, one of the interesting things that's been shown in the last few years is that microarray studies, SNP chip studies, often greatly underestimated the variation across wild populations, and even to the point of completely getting the phylogeny wrong. So that in the pigs, for example, a lot of the pig trees that were built using SNP chip data made it look like they came from Europe and not from Asia, because all the SNP chips had been designed against European populations. But when they started doing just unbiased genome sequencing, it turned out that the ancestor was in Asia, as was previously thought, because they'd been missing all of that crucial Asian variation. So that's definitely a problem, and it is necessary to think very carefully about that bait design. And it might be worth developing sequence for a distant species as well, and sort of designing baits in between.
Okay. All right. Okay. ¿Hay alguna pregunta? Well, we appreciate very much the webinar. I think the next one will be on the 14th, or is it 28th?
The next one is hello.
Thank you very much.
Thank you.
It's the next one is October 28th.
Okay.
So we'll see you then.
All right. We expect the announcement soon. Thank you very much.
Okay, great.
Bye.
Bye.
Bien, entendieron bastante.
He's asking if there's so much time.
Nice job. great talk.
Yeah, that was really great.
You think?
Yeah,
Can I still?
Yeah,
really great.
Thank you.
I don't know if we're still.
Yeah, I think we have more
maybe
people this
still
time
here.
around.
Okay, that's good.
But I don't see professors, which I would like more faculty.
Can they still hear us? Oh, yeah.
I think might be able to hear us.
Let's make sure it's
Yes, so you